Great to see you all. What a great turnout tonight. My name is Caroline Jean Fernald. I'm the Executive Director of Harvard Museums of Science and Culture, a partnership of 4 public-serving museums where our mission is to connect collections and research at Harvard with global audiences, fostering community and deepening appreciation for science, the natural world, human cultures, and our shared experiences. With this mission in mind, I am delighted to welcome you to this year's John M. Prather Public Lecture. HMSC's partnership of museums crosses many disciplines, and we host a wide array of events throughout the year at our 4 museums. On April 21st, the Harvard Museum of Natural History will host the conversation, Whose Genes? What Genes Can and Can't Explain About Life, which will address one of biology's most enduring and contested questions, how do genes shape organisms, identities, and evolution? On April 23rd, the Peabody Museum will host, and I love the title of this talk, From Spam to K-Pop, the Korean War and the Rise of Modern South Korea. Any lecture that brings up spam. Based on photographs taken during the Korean War, the lecture explores how this time of conflict influenced South Korea's rise to an economic powerhouse and global cultural trendsetter. On April 28th, the collection of historical scientific instruments housed in this building will host the panel discussion, Making a Nation, Technologies of the American Revolution, which will highlight the role of surveying, electricity, and mathematics in the Revolutionary War and its wider historical moment in the Boston area. To learn more about additional upcoming museum events, I invite you to visit our website, hmsc.harvard.edu, where you can sign up for our newsletter, or you can also follow us on social media and all the usual channels. We are grateful to all our members and supporters for making these programs possible. It is now my pleasure to introduce Stephanie Pierce, Professor of Organismic and Evolutionary Biology, Curator of Vertebrate Paleontology, Interim Curator of Mammalogy, and Alexander Agassiz, Professor of Zoology in the Museum of Comparative Zoology, who will introduce our speaker. Thank you.
All right, so thank you, Caroline, for the introduction, and I want to say good evening to everyone here today. So it's my pleasure to welcome you to the 2025-2026 John M. Prather Lecture Series. The Prather Lectures represent our most distinguished lecture series, organized through a long-standing effort between the Departments of Organismic and Evolutionary Biology and Molecular and Cellular Biology. This series is made possible by a gift from John M. Prather, a member of the Harvard class of 1894. Dr. Prather was a dedicated zoologist and educator who spent much of his career teaching science at the high school level. Today, his legacy continues to support the mission of bringing groundbreaking biological research to the Harvard community and to the general public. I also want to thank the Harvard Museum of Science and Culture for co-sponsoring this evening's lecture, as well as Nicole Adams and Professor David Haig for their hard work in helping to organize this year's Prather. So now it is my honor to introduce this year's speaker, Professor Neil Shubin. Professor Shubin is the Robert R. Bensley Distinguished Service Professor of Anatomy at the University of Chicago and President-elect of the National Academy of Sciences. For Professor Shubin, this evening is a homecoming. He earned his PhD here at Harvard in Organismic and Evolutionary Biology and the Museum of Comparative Zoology in 1987. It was during his time as a doctoral student that he cut his teeth in paleontological fieldwork, springboarding a career in discovery-based, curiosity-driven science. Professor Shubin is globally renowned for his fossil discoveries, most notably the 2004 find of Tiktaalik rosier. This fishapod serves as a critical intermediate in the fossil record, representing the transition between life in the water and life on land. However, what makes Professor Shubin's work truly transformative is his multidisciplinary approach. He doesn't just merely study the fossil record to understand how the fish body plan transformed into the tetrapod body plan. He bridges this with evolutionary developmental biology. By experimenting with embryos of species like sharks and paddlefish, his lab pinpoints how the genetic blueprint of a fish was tweaked at the molecular level to form limbs with fingers and toes from what were once fins. Through his best-selling books, such as Your Inner Fish, and his award-winning PBS series, he has made the deep evolutionary history of animals accessible to millions, demonstrating how every organ and gene, even in our own bodies, preserves traces of our fishy past. Ultimately, Professor Shubin's career epitomizes how digging in dirt can lead to a phase shift in our understanding of the evolution of life on Earth. So please join me in welcoming Professor Neil Shubin to his lecture, Your Inner Fish Revisited, New Insights into Our Fishy Past.
Thank you. Thank you, Stephanie. Thank you. Thank you so much. Today we're going to talk about new discoveries that help us explore our fishy past. But first let me say that's going to be a long story that's going to span 40 years. But my own origin story, in fact, where I got locked into this problem, including the whole conceptual and cognitive approach to deal with it, really began here. And by here I mean Harvard University and its special intellectual environment. By here I mean the museums at Harvard, which infused in me and generations of other students the tools to make discoveries in the fossil record and beyond. And here in this freaking room in the back where I was a teaching fellow terrified in my first 4 years of graduate school. So it's here in a lot of deep levels. But there's another way that this all began, and I want to sort of begin at the beginning for me, is where so much of science is finding the right question, right? And then tethering it to the tools to answer those questions. And for me, the question began in a class at Harvard University in the beginning of my second year of graduate school. I was a nervous second-year graduate student because I hadn't yet discovered what I wanted to do for my PhD thesis. And my mentor, the person who became my mentor, the late Farish Jenkins, who you'll hear about throughout this talk, Farish led a graduate seminar on great transitions in the evolution of vertebrates. And each week was a different transition. And each week a student had to present that transition. And it was like speed dating with the great concepts in our field. And I remember Farish would pull out these very simple cartoons that told elegant stories. He really was a very visual instructor. And this was one that was actually produced by Sharon Emerson for a textbook in the mid-1980s, which was, we knew a lot about the fish to tetrapod transition, the invasion of land by fish. But this sort of summarized it in a cartoon form, very simply for an introductory class. And I remember looking at this, and you see a cartoon of a fossil fish on top. That's a cartoon of a fish that first appears in the fossil record about 390 million years ago. That's 390, a long time. And then the early tetrapod on the bottom, which we had several fossils, and that's a cartoon of a creature from rocks in Greenland, from rocks about 365 million years ago. And I remember looking at this diagram thinking, fish live in water, tetrapods on land, or at least quasi on land. That's a first-class scientific problem. How did fish evolve to walk on land? Because everything has to change. The global ecology is changing, function is changing, anatomy is changing. The genes and developmental processes that drive much of this are changing. It's a wonderful problem to approach from a multidisciplinary viewpoint. And so really, that was 40 years ago, and I'm still thinking about this thing. It's kind of, it's really something how that ignited in me. And the other thing it ignited in me personally, but other students at the time, was that this is an answerable question by different empirical approaches. One is developmental biology, which I'll talk about on Friday. But today I'm going to really talk about the paleontological piece, which is what I came to Harvard to study. And in that piece, it was very explicit to find new fossils, to find new places that hold new faunas that tell us about great transitions in the history of life, whether it's the origin of mammals, the origin of birds, the origin of tetrapods, and on and on and on. And so I was approaching this originally as somebody thinking about expeditions, where to lead expeditions to discover new evidence that tells us about this great transition. Can we find a flat-headed fish with fins, with arm bones inside, something intermediate? Well, the paleontological playbook for that kind of thing is extremely simple conceptually. It's really hard in application. And that is, you look for places in the world that have rocks of the right age and rocks of the right type to hold fossils, rocks of the right age to answer your question, rocks of the right type to hold them, and then rocks that are exposed to the surface. I mean, that's it, conceptually. It gets a lot harder because you're making tons of estimates about age and location and so forth. But I think in looking back on it 40 years later, why am I saying 40? That's a long time. 40 years later, the real breakthrough for us came from placing a bet. And that bet was that if we focused on places in the world that had rocks that were formed in ancient delta systems, where we had rivers and streams and meandering plains and estuaries and nearshore environments, that package of rocks that were contained in those environments, if we had places where we had that package of rocks, we're in the game, right? We're in the game because you can sample ancient tidal flats, ancient estuaries, ancient oxbow lakes, and on and on and on. And looking back, it was really kind of identifying that. And the other thing that made that very achievable is it's very identifiable in the geological record where you have this kind of, it's called clastic wedge, that kind of thing. So that really became the target of my hunt, finding rocks of the right age. In this case, it's late Devonian, middle to late Devonian, and rocks of the right type that were formed in these deltaic environments, ancient rivers, streams, nearshore environments. That was it. There was a fourth criterion, and that was my lack of money. And that's where I began my career at the University of Pennsylvania. My first 10 years of my career were at Penn. And it turns out Pennsylvania was kind of perfect. And I'm showing you kind of what's important about the state, other than Philadelphia, Pittsburgh, and Harrisburg. But what you see is in purple here, where there's mapped Devonian age rocks. And those Devonian age rocks are the right age. Also, that portion of Pennsylvania was an ancient delta system. So I had an ancient delta system of rocks of more or less the right age, about 3 hours from home where I could just drive. And I had a new graduate student, Ted Deschler, who I've since worked with, along with Farish. Those two, Ted and Farish, are really my co-conspirators in much of what I'm going to tell you for the first half of the talk. So we began that, and it began by just driving around Pennsylvania and following the Pennsylvania Department of Transportation around as they built new roads. Because when PennDOT decides to do a new road, widen a road, put a bend in a road, they blow up a lot of rock, hopefully, and hopefully that's Devonian age rock, and we'd have stuff to look at. And this is one of the great road cuts that we worked on. It's about an hour north of State College, Pennsylvania. There's a human being for scale right there. There's our cars. So it's a big section of a slither of time in the Devonian, where essentially those are all cross-sections of ancient rivers and streams, meandering rivers and streams. And there are all kinds of fossils in there. As soon as we got there, we started to find fossils. I mean, it was really amazing. And in fact, it was not too long before we found early tetrapod fossils. Arms, legs, shoulders, skulls, pelvies, fingers, toes of some of the earliest creatures to walk on land. And this is all by the side of a road in Pennsylvania. And what I love sharing with students is that's just, when you're thinking of geology, this juxtaposition of present and past is really kind of awe-inspiring in its own way. Here I am in central Pennsylvania by the side of a road, but I'm digging out fossils from ancient ecosystems that were tropical and ancient, fish and so forth. And we're able to pull out, reconstruct, Ted and I, with National Geographic, what these Devonian streams looked like in the late Devonian. You had some of the earliest forests with deep root systems, which is a big deal. You had all sorts of invertebrates on land. And in the water, you had it was really just loaded with life. 15-foot-long predator fish with teeth the size of, your thumb. So you wonder why tetrapods left the water. Hello, that. You have all kinds of tetrapods, limbed animals. You had armored fish. I mean, it was a real cornucopia of Devonian life. And it was a really wonderful site. We could have spent a long time there working it. But Ted and I realized we had a problem. And that problem is we were in the wrong age. We were in the right rocks. All right, so proof of concept, that sort of bet on the rivers and streams and these delta systems, A+, that seems to be working. C- on the age. Why? Because we're finding really highly derived tetrapods. We were already, to go back to the motivation diagram, we were finding a lot of derived tetrapods. We were in rocks about 365 million years old when we compared it to other places around the world, and we mapped it on a phylogenetic tree, an evolutionary tree. It was very clear we were probably, almost certainly in rocks too young. Probably about 10 or 15 million years too young when we looked at other faunas from around the world where scientists, paleontologists collected fossils. And where we needed to be was this is going to make sense in a second, in a portion of the late Devonian, a stage known as the Fronian. The S is silent. It's a time window around 380 to 375 million years old. That's when we needed to be. That's where our bet was. So back to the drawing board, now with a little more confidence. And we were looking around for where we'd have these deltaic rocks of the right age, in this case, Fronian. And everything changed when we ran into Dott and Batten, a classic geology textbook. Some of it was pre-tectonic, plate tectonic. And there was a set of diagrams in there that were very simple, but they kind of embarrassingly pointed out what I didn't know. And that is, here's a map. This is from Dott and Batten, the textbook, actually, a college geology textbook. Inspiration's where you find it. Here's North America, Central America, and superimposed on that is an interpretation of the environments that the Devonian rocks were formed in. And so they modeled here as you had sort of ancient marine sediments, Devonian age in the western part of North America. But then they identified 3 areas shown in red, and I think you see where I'm going, that were formed in ancient delta systems, had a series of references. This is like 1998, where you would go to the library to find these references. You know, that place where they had paper and you cut your finger and all that good stuff, that was where we went. And so we knew this one, it was Catskill Formation. That's the Catskill rocks, been there, done that. We knew this one, Greenland, because colleagues working, you know, from Swedish colleagues working in Greenland in the 20s and 30s discovered that tetrapod had been showing in the cartoons so far. They discovered that in Greenland way back, along with other critters. But I think you see where I'm going. Extending 1,500 kilometers east to west was a bunch of Devonian. It had a series of references pointing it out that were mapped as Devonian age, formed in ancient rivers and streams. I looked at Ted. I said, do you know anybody who's worked up there? He said, I don't know anybody. Do you? I said, I just asked you that question. Nobody worked there. So we ran to the library. And it took us a while to figure this out. But this was the scientific paper that I point people to say, whenever they ask me, how do you know where to look for fossils? I point them to Ashton Embry and Edward Clovan's Middle Upper Devonian Clastic Wedge of the Franklinian Geosyncline. Scientific paper. I don't think it's going to be a Marvel movie, MCU movie, or anything like that. But there's one page in this diagram that kindled decades of work in the Canadian Arctic. And that is when they talked about the age of the rocks. I think you see what's happening here. The available data indicate an age of early to middle Fronian. Thank you very much. And then when they talked about the environment, what those rocks were formed in, this is where I literally got goosebumps. The Fram Formation, which is the name of those rocks in the Arctic, is similar to the Catskill Formation of Pennsylvania. Here we had rocks similar to the fossiliferous units of Pennsylvania of the right age. So that's kind of what launched years of these expeditions. That's why we sat on it for so long, because we were so confident that it would have, we knew it would be a long-term project, but that's why we sat on it. So I'm just going through the story. Now, instead of driving a station wagon to central Pennsylvania, you now have to leapfrog beyond a tank of gas of a helicopter, and you have these aircraft known as Twin Otter aircraft, which can bring the fuel in. We had to bring a crew of 6 people to remote places for a number of years, and it took us 6 years to be successful initially here. So it was not something that happened easily by any stretch of the imagination. So this is kind of the diagram that kind of told us where to look, and then we refined it more with geological maps. But here's where it's circled in red are where the middle upper Devonian rocks are at the surface. And you can see it's a vast area when you look at the scale of the thing. It covers, now we had a different problem. Where do you work in the Canadian Arctic? It's a big place. The fossils are small. How do you do that? Now it's a whole other layer of filters we needed to use. And I want to work you through that a little bit to show you how we think. So we were all excited. We started in 1999. Took us a while to raise some money. Took us a while to get some permits. So we started in the western part of the Arctic. This is one of our first camps in 1999. These personal tents with a wind wall will stay up in 70, 80 miles an hour. This kitchen tent was our first one, would stay up to about 25 miles an hour. So I used to chase, Farish and crew, we'd chase this thing all around. But anyway, in a storm. But this was kind of a failure, but it was an important failure. Number one, it taught us how to work there. Number two, it told us where not to look. These were marine rocks. These were rocks formed in ancient oceans. Clearly not where these animals were lived. We made a bit of a mistake in selecting the camp. Furthermore, this is a place where the weathering conditions, that is how the rocks erode, were not conducive to finding the kinds of fossils that we were looking for. We were used to working in the south, where you didn't have the freeze thaw you have up in the Canadian Arctic, or actually in the Antarctic where we worked later. And so that changed the weathering conditions a lot. So we learned about the weathering conditions. We learned about the topology. So failure, but lessons learned. So we went back. And when we went back, I'm using a diagram of Pennsylvania, that's the Catskill Sea, but this applies to the Arctic as well. We wanted to go upstream in the ancient world. So we were in the middle of the ocean. That meant, in this case, meant going east, following the geological map. So we went east in 2000. Boom, there we are in southern Ellesmere Island. New kitchen tent, thank you very much. And new erosion regime. More cliffs, more traditional fjord, more sort of high angle of repose, which actually gave us fresh rock to look at. Gave us other problems, but gave us fresh rock to look at. And here we're getting closer in 2000, each year a little closer. In 2000, we started to find bones of lobe-finned fish. We were in the right environments. But these lobe-finned fish, they were finding the bones were bits and pieces. They weren't skeletons. They weren't going to do much. Bits and pieces were fine. We're on the right trail. But it turns out that the rivers and streams contained in these ancient Fronian Age Devonian rocks were high energy. They were rapidly flowing. They had big pebbles, and they were containing lots of detritus in their beds. And bones were part of that. So these were all highly weathered. We needed a place that had more gentle flowing streams, that had streams where we can sample what's known as the overbanks, the margins of those streams. We needed something, right environment, but we needed a better setting to find whole skeletons. Well, that brought us to this place, which is further east in Ellesmere Island. And this is the most remarkable slide in my collection. And I didn't take it, and there's a reason for that. So Ted took this shot. I think he took it because he just finished lunch and he liked the view, this little gate here. But what he captured is a blue pixel. There's a blue pixel right there. I don't know if you can see it. That little blue pixel is young Jason Downs. Jason Downs was a college student who joined us on the expedition. And Jason, just like Ted, finished his lunch, didn't know this at the time, but then pulled off the screen here, walked away. All right, so Jason walks away. Now, big mistake. Jason was alone. He shouldn't have been. We usually walk in pairs. Jason somehow got separated, but he walked off the thing. Anyway, the day goes by. I was somewhere else. And went back to the main tent that night around 7 to make dinner. Ted comes in. We're making dinner. We're chewing the fat. I said, you see Jason? I said, all right. I said, Jason, you see Jason? I just asked you that question. Where's Jason? All right, so like if you're a faculty member, right, in the faculty handbook, there's clearly, they don't even need to say it. It's like, don't lose your college students in the Arctic, all right? And I just lost a college student in the Arctic. It was a big deal, all right? And I was, that's, it's bad news. We don't like to do that. So anyway, Jason comes ripping into the tent, and in his parka, he's pulling out bones. He says, I found it. It's like, what'd you find, a polar bear? No, we found all kinds of lobe-finned fish bones, bits and pieces. And here's what's happened. This is why this slide is so remarkable. Jason there, that pixel, Jason walks off the slide. Then later in the day, and the reason why he was late back to camp, is he walked over here. You can see it's like slightly different color. It's kind of gray. That's because it's a carpet of fish bones. I mean, thousands and thousands of fish bones. And he was like wide-eyed, right? And so we were like, Jason, where's this lair? It's about a mile from camp. So dinner can wait. We grab some Cliff Bars. This is us at 2 in the morning because it's daylight 24 hours a day. Crawling Jason's site to find the lair that those bones come from. And it took us, I know this is a saga, it took us another year to find that lair because remember I told you the weathering conditions? Just getting the exact lair turned out to be really tough. But find it we did. It took us another year. This is by 2002. We started again in 1998. This is the layer, Jason's layer. There's Ted there, big old smile, because he has the layer here. And as soon as we hit the layer, that carpet of fish bones was produced by skeleton after skeleton of fossil fish piled on top of one another in the layer. And they broke up as they weathered out, and that's what produced that carpet. So now our job came to let's work this layer. So another year. Working that layer, and we'd kind of traditional quarry. We worked the layer, be all next to each other, 6 of us. And we opened up a big hole. We worked the layer. If you were to visit there right now, it's what it looked like in 2006. Not anymore. We'd work next to each other and finding bones. Not really much anything new until, this is Farish right there. Sorry, Farish, at your back. Steve Gatesy, who was an emeritus professor at Brown, pulled a rock from this little spot right there. And he pulled a rock, and he pointed to this area. He says, Neil, what's this? And as soon as I saw that, I knew we had found what we had spent 6 years to that point looking for. Now, you think I'm crazy, but this is it. Look at this V. See this V right there? Those are two jaws upside down looking out at you. There were teeth here that I could see the teeth gleaming. And furthermore, there was a snout on the other end. So it's an upside down skull with a snout on the other end. And it's not just any snout. It was the snout of a fish, because I could tell by the texture on the bone. And the shape of the snout suggested it was flat-headed. You know that cartoon I've been showing you with the flat-headed? We had a flat-headed fish staring at us. That was a good day. So we removed this, and it comes home at the base of a helicopter. There's a first-year graduate student for scale. That's the fossils in there. We love our students, though, for they love doing that. I don't know why. I'm too old to put a hook on the bottom of a helicopter, but they love that. Anyway, it comes back to the lab. Now, we've since found 20 of these things, so they're not remotely rare. But this one was very special, and as soon as we removed it, we found 2 more of these skeletons of this fish, Tiktaalik. And then that particular one came back to the lab. It's now known as the type specimen. And this is it after 5 months of preparation. You can see it's like on top of a head. There's one orbit, there's another, like you're seeing that emerge. Another 5 or 6 months later, I'm forgetting the time period, but there, look, flat head, eyes on top, clearly fish ornament on the bone. There's one shoulder, there's another. There are no bones like operculi and so forth connecting it to the shoulder, so it's clearly a more tetrapod than fish, but maybe it's a fish because the bones are kind of crazy that way. So, this is one of those stories where, when I was out in 2000 after the discovery of this critter, we were looking for an intermediate, maybe a flat-headed fish with fins with arm bones inside, went to places in the world that had rocks the right age, rocks the right type, rocks that were exposed to the surface, and this is what we knew of Tiktaalik rosea in 2005, published it in 2006. And if I was to hold this particular specimen up for you, this one's about 4 feet long. It gives you a scale. Flat head, eyes on top. Like a fish, it has scales in its back and fins with fin webbing. Lots of other fishy characteristics as well. There's a gill skeleton in there as well. But like an early tetrapod, it has a configuration of bones that are like a land-living animal, and inside the fin are bones that correspond to upper arm, forearm, even parts of a wrist, and as well as other features as well. So it really tells us a lot, about the transformation. Now, we published on it. And I'm giving you the discovery story today. Right now, we're at 2006. There's more to go. Sorry about that. What do you notice about this diagram? It's like a pinup from the 1950s with something strategically hidden, right? Only the front end. What's going on back there? You notice how we have this thing coming out of the mud? There's no back end there. That's because we didn't have the back end. And so, now we had a new quest. We got to find the back end of this thing. But it turns out we did have it. So this is the ventral surface, the belly surface seen in ventral view of that type specimen of Tiktaalik as it was in 2006. Turns out we brought another block back with us that took 5 more years in the laboratory to prepare because it was very thin and really involved some very special, very fine work, but it was there. And in it was parts of a hind fin. So we did have it. And in fact, indeed, there was a pelvis in there as well. So cutting to the chase, the lab work, so it's a little more boring. I'm not going to show you all the slides of, hey, here it is on April 1st, and here it is on May 3rd. But I respect you a little too much for that. But but essentially what it did was, here's a fish like Eusthenopteron. That's that cartoon of a fish I've been showing you earlier. These are different flavors of Devonian tetrapods. This is Tiktaalik. Essentially what you're showing is we had the pelvis, and you could see in the evolution of the pelvis in the hip here, you go from something relatively small to something very big, which is shared with tetrapods. So it's emphasizing the hind fin in some ways. But it's still very fishy. Its shape is very fishy. I mean, there are things that are derived about it, but it's still kind of it's big, but still very fishy. And you can see the shape of the ribs and the regionalization of the ribs and the size of the ribs and all kinds of other things. But this created a new problem for us that really bugged me. This is the pelvis and ribs of Tiktaalik. And there's clearly, I'll show this in more detail tomorrow, but there's clearly a contact between what's known as the sacral ribs and the ilium here. There's actually a joint preserve, so there's support for it, and it's big. But what you'll see is when we tootle through this thing, the hip joint is huge. Here's the acetabulum, it's the hip joint, socket. Look at that socket. See that socket? It's facing downwards. But it's a giant socket and a giant pelvis. That's doing something, right? So this is kind of so we scanned every bone of the type. We scanned every bone of Tiktaalik, by the way. You can access all the bones and print them out in any color, any size. of the type in every other specimen we've collected. But this was it. And I remember thinking, this is how simple my life was in 2019. This little gap here is where the femur would be, and we didn't have it. And that really pissed me off, okay? I didn't have the femur. I had this amazing hip joint, but I had no femur. I didn't know where it worked. And my life was so simple then. I look back and how can I pick now? It's like I'd be like blissful thinking about that's all I worried about in 2019. Anyway, there you go. That's the statement of the world. But anyway, it bothered me. I didn't have the pelvis, okay? I mean, I'm sorry, I didn't have the hip joint, the femur. We didn't have the tail either, but I had no sense we'd ever get the tail, because these are fluvial rocks formed in rivers, and everything's kind of beaten up a little bit, and the tail's very thin, so I didn't have hope. But I had hope that Tiktaalik was common enough. Maybe I'd find a specimen with head, shoulders, hip, heads, fingers, toes, so that sort of thing, the little whatever. All right, so this thing was my nemesis for a while. So we tried to get back in 2019, and we got the permits, we got the money. Then COVID hit in 2020, couldn't go. Arctic was closed for fieldwork in 2021. It was closed for fieldwork in 2022. We went up in 2023. We're finally excited to do the femur hunt. We got up there. We shipped all our gear. Nobody was hurt, but the helicopter crashed. Had to go home. Never being in the field. That was not fun. And then in 2024, we finally got to go back. Now, where did we go? So this is Ellesmere Island. So the Tiktaalik site, the original Tiktaalik site is where the tip of that arrow. But Ted and I, for years, wanted to visit this fjord right here, Goose Fjord. And the reason for that is to get to the Tiktaalik site, we'd fly over this patch of Ellesmere Island. We knew it also, its geology from the geological maps and all that good stuff. But we'd see this thing, and it's beautiful exposure. It doesn't look beautiful, but it's really beautiful exposure of Devonian rocks. This is taken from trying to get to the Tiktaalik site in 2006, but we took a picture of this. We said, gosh, we got to work there. If we ever got to go back to find more Tiktaaliks, let's go there. And that's exactly what we did. So we went back in 2024. We tried 5 years to do it. After a helicopter crash, we finally got back. And this is it in that year. It was beautiful when we landed. You saw that snow in the previous aerial photo? Look at this. This was it in July 7th, 2024. We had personal tents, new fancy kitchen tent. It was perfect for about a day. And then the storm started to come, and we got hit by cyclones that kind of came right up this valley. And one of them took this tent and blew it a mile away. A mile away. And all the food and stuff that was in that tent blew that about half a mile away in the middle of the storm. So we had to run and get all that food. And so we're scurrying around down this little hill here, picking up all our food in the middle of this storm. And I look up, and I see one of my colleagues' tents 20 feet in the air. It was 80-mile-an-hour winds that ripped it out. It was a bad day. But the day before that storm hit, I kid you not, was one of the best days. Because walking back to camp over a site that we wanted to look at very carefully, we found on the surface, no digging, Tiktaalik pelvis, Tiktaalik shoulder, front of Tiktaalik skull, Tiktaalik forefin, and guess what? Two pelvies with two Tiktaalik femora. And I just so happened to have a graduate student, Emily Hillen, who was with me, who was working on the pelvis of Tiktaalik, and her thesis became the hip joint of Tiktaalik, because she's holding two femora that were found on the surface. And that's Emily with a big old smile right there. And these were found on the surface, and I'm going to show it to you as it was collected. It's like never happens, right? This happens. This is camp the day after everything got lost. So the kitchen tent's way up the fjord here. Those are the three remaining tents. That's that one that blew in the air. You get the idea. We pay a price for these bones, trust me. But anyway, so you're going to see the femur of Tiktaalik spinning around. We haven't published on it yet, but this is kind of what it is. But you can see there's the hip joint right there. And it's mostly ventrally facing, because that's the ventral or belly side view. But like you can see, it's beautifully preserved. This was prepared not by humans, but by the wind in the Arctic. I mean, that's amazing. And look at that. That would fit into that socket of the hip. And you could see by its orientation how it would lie, facing downwards eventually. Pretty remarkable stuff. But it gets better. We have the knee of Tiktaalik down there. But let me show you what's interesting is when you take this is the articular view of the femur of Tiktaalik, and this is the articular view of the pelvis of Tiktaalik. Look at that. There's that extra facet there, extra little space. There's that whopping big acetabulum. That's what motivated the 5 years of pain looking for the femur. There's a big head that fits in there, but there's another head. It's bicondylar. It's got two heads, such that when we orient them together we can do this quantitatively by matching maximum surface areas. What you have is a situation where this thing locks in. Now, it can move like a fin. It's very much a paddle, the hind fin. It can move like a paddle because it has a very mobile socket, that acetabulum. But when it goes ventrally, it can lock in place with this extra joint that prevents it from moving forward, that locks it in, so it can serve as like a strut or a prop or support it for swimming. But you're getting what's called, you're getting passive stability from the skeleton. The skeleton can support it without muscles necessarily doing the entire job. And so it was pretty fun, because now I'm no longer pissed, well, I'm pissed about other things, but I'm no longer pissed off about the femur, because it's telling us, I mean, many beautiful things about, quadrupedal. I mean, what's going on with the hind fin? This is a fish that did not skip leg day, okay? It's got a big hind fin. It's got a huge pelvis and on all good stuff. And so so we can talk, and I will tomorrow, about the fish to tetrapod transition in much more detail. But really kind of what inspired inner fish, and when we think about our fishy past, is our connection to the rest of life on our planet. Because as Stephanie said in introduction, in every organ, in every tissue, in every cell, and in much of the DNA of our bodies, we contain artifacts of the history of life. And some of it goes deep in time, to billions of years. And we know this because we can collect fossils that connect us to the rest of the world, life on the planet. We can look at embryos, we look at genes, and on and on. And when you think about Tiktaalik and its evolutionary cousins, we know other creatures like Tiktaalik existed here. They're known in the fossil record. There are several other genera. This is not a one-off. There are other creatures there that tell the story as well. But when you look at Tiktaalik and its evolutionary cousins, it has features that we see for the first time that become parts of our own bodies. These wrist joints, which we see in Tiktaalik and its evolutionary cousins that evolved in the Middle and Late Devonian, you can trace it, and you can see they become our wrists. You can trace the humerus from Tiktaalik, from primitive fish to Tiktaalik, to amphibians, to reptiles, what we call reptiles, other mammals and people. The elbow, the hip, on and on and on. We can connect our anatomy to other living creatures, other vertebrate forms, by collecting these fossils. And what that means is the fish to tetrapod transition, when we can do this, isn't some esoteric side branch of evolution. It's a part of us. Every time you bend your wrist, every time you shake your head, you can thank these fish evolving in Devonian lakes, streams, tidal flats, 380 million years ago. It's a story we see in fossils. It's a story we see in embryos, when we compare embryos of different species. And it's a story we see in the genes that are behind the embryo, that drive embryonic development. And I'll talk about that almost exclusively on Friday, but I'll do a little bit here. And so, when you work on fish, there are fish people here, I see fish people here, you get a lens of the world, right? It changes how you see the world. You normal people, most of you normal people, look at this and you see Albert Einstein, really smart guy, a pinnacle of human achievement. I look at this and I see a big bipedal, big-brained fish. And when you compare Professor Einstein to the fish, thank you, Einstein's on the left, I labeled it for the fish people here. So, you can compare not only his anatomy and connect it, but you can look at development. Look at Einstein's head, look at your head, look at my head a few weeks after conception. This is kind of a cartoon of what an embryonic vertebrate head, mammalian, particularly human head, looks like. You have paired primordia for the eyes in the front here. You have a series of swellings that go from front to back, which I colored in. 4 of them here. Other types of species have 5. But you can see little clefts and so forth. And when you look at fish, when you look at turtles, when you look at other vertebrates early in development, their heads don't look identical to ours by no stretch, but the embryonic heads have a certain roadmap to them. Paired primordia for the eyes, and these paired swellings known as the pharyngeal arches, which go from front to back as I color-coded them. And they have cells in there. And those cells are dividing in place, and there are other cells that are migrating, and they migrate in there, and they do all kinds of stuff. And you can trace what those cells become in development. And in a shark, that first light blue one becomes portions of the upper and lower jaw. The second one becomes gill bone, a set of bones along the gill rect there, and others become portions of the gill apparatus. But this is not just the bones, it's the nerves and arteries and muscles. They're all sort of developing from those cells and that sort of pharyngeal organization you see in the embryo. Logical question to ask, what's going on with mammals and like humans? That first one becomes portion of the lower jaw and 2 bones in the middle ear. The second one becomes a bone, the hyoid bone in the throat, and 1 bone in the middle ear, including a bone of styloid process at the base of the skull. Another one becomes part of the hyoid apparatus, which is, again, that throat bone connected to the tongue, and then the voice box. Now, what does this mean developmentally? What this means developmentally is many of the muscles and nerves and bones I'm using to talk to you right now, and many of the muscles and nerves and bones you're using to hear me with correspond to gill structures in sharks and fish. Now that seems odd, you're comparing development, but if I look at fossils, I could trace where this bone here, this gill bone, the hyomandibula, it evolves in fish, gets smaller and smaller until it becomes the stapes in the middle ear. So development makes a prediction that what we'd find in the fossil record when we compare it, and it confirms it. So we have multiple lines of evidence that work together to give us this beautiful story of the part of the fundamental organization of our skulls. And when you think of our fundamental organization, you think about the single-celled egg, which contains proteins, RNAs, and DNA, which contains information that will unfold during embryonic development, in interaction with the environment. All of us began as a single cell, a fertilized egg, but we have 20 trillion cells, 25 trillion of our own cells. There's tons of microbes, more of them in our body, and much of our evolution and development is dependent on that relationship. But we call that process going from 1 cell to 20-some trillion cells, all packed in the right place. That's development, right? That's bodybuilding. So the problem of development is going from this single cell to this thing, right? With lots of cells all differentiated, packed in the right place. How is that information encoded? How does it unfold? And once we understand that for one species, what does that tell us about how they evolve? How the sort of the DNA toolkit that builds this body, how did it evolve from other creatures? And that's kind of the challenge and excitement of developmental evolutionary biology. And a lot happened starting in the 1980s, which really changed my roadmap to being a comparative biologist, where it's discoveries like this, looking at an embryonic fly, right? And looking at genes that are active during the development of an embryonic fly. And the genes are turned on in a regular, these genes, Hox genes, are turned on in a regular pattern in the fly body, along with other genes that come out before them. But these are involved in the correct placement of organs and the basic body architecture seen in flies. And turns out, what was the revolutionary discovery that really changed the way we think about this empirically is that other animals have versions of these genes too, including frogs, including fish, including mammals like humans. And what are they doing? They are determining portions of the body axis, correct placement of the limbs, differentiation of the vertebral column. That is the basic bodybuilding toolkit. There's deep conservation in some aspects, not entirely, some aspects of that, which was a real roadmap. And these genes are not just active in the body axis, they're active in developing limbs, genitalia, nervous system, and so forth, such that in my lab, for instance, we've been able to look at them, the one gene called HOXA13, we're able to look at it in mice, and this is it mapped in terms of what's shown in green here, is where this gene is active in an embryo and where those cells end up in an adult. So this is an adult mouse, and it turns out these genes actually mark the whole wrist and digit area of the mouse limb, such that if you construct a mouse, which was done by other labs, that lacks these genes in the limbs, it's missing this portion of the limb. Do fish have the genes that mark our wrists and digits? Yes. What are they doing? They're marking the terminal end of a fish fin. So when we do those same experiments on a fish fin, and when we knock those genes out, when we modify them, we're finding that they function in very similar ways, in different contexts. So this is kind of your inner fish seen in the genetic level as well, your inner fish fin in your wrist. But you can also take this a little bit deeper, and I'll end with this sort of side branch example, but I think it's relevant as well, and I'll take your questions, is if we look at the earliest fishes, things like these jawless vertebrates, you start to find skeletons of these rocks about 450 million years old. And these are early jawless vertebrates. They got no jaws, but they have these exoskeletons composed of these discrete sorts of scales. when you look at the scales on the exoskeleton of the earliest vertebrates, there's something pretty remarkable about them. They got something like this. You can pop them in a synchrotron, high energy light source, get a sense of their histological cellular structure. You can do all that good work. And you start to see they have a little structure in them. They got tubules, they got a covering. In fact, when you look at them, these 450 million year old scales, they have an outer layer, they have an inner layer, they have a cavity. There's all kinds of stuff going on here. In fact, when you look at the materials, they have on the outside, they have enamel, hydroxyapatite, pure hydroxyapatite, enamel, kind of like you know where I'm going with this. They have dentine with tubules in there. They have classic sort of structure, very similar to something in our own bodies, and they have an interior of bone that holds it together. When you do the comparison, it turns out these early scales are essentially identical in many ways, histologically, to modern teeth. So what are our teeth today originally formed as part of the exoskeleton of early jawless fish. And in fact, when you look at these structures, teeth are highly innervated. You know that when you go to the dentist, right? Not pleasant. These are innervated as well. And in fact, when we look at analogous structures, or actually homologous structures, these kinds of scales are present in some living fish, like catfish and sharks. They're highly innervated. They function as a kind of sense organ. So this is not my lab, but this hypothesis that's really been supported very much is the outside-in hypothesis for the origin of teeth, that teeth originally arose on the outside of the body in the exoskeleton to become our teeth. So every time you go to the dentist, yes, you can not thank, you can thank your inner fish for your wrist and your neck, but you can also curse your inner jawless fish for the pain you feel, because that originally involved the sensory structures in the exoskeleton of early vertebrates. So you can ask the question, you could say, like, okay, I get it, well, interesting stories. Who cares about your inner fish? But the reality of things like your inner fish is utterly trivial to a working biologist. Because what do we use for our biomedical models of human disease? We use fruit flies, we use fish, we use mice, we use yeast, right? And you think about the Nobel Prizes. 2 Nobel Prizes is awarded to 5 people in the last 17 years of going to folks working on Caenorhabditis elegans, right? A tiny little worm the size of a comma on a piece of paper. Yet that's providing insights into how our genes are silenced and how our cells are programmed to die naturally. And that's just the tip of the iceberg, right? When you think of studying other creatures and you think of CRISPR-Cas genome editing, this technology that's revolutionizing healthcare with modifications of it, that began by people studying the bacteria and microbes in salt marshes. When you think about GLP-1 receptor agonists, Ozempic, Wegovy, and all that, a big step in the evolution of that therapy was people working on Gila monster venoms. That is curiosity-driven research about that sponsored by Harvard and the Museum of Comparative Zoology and other institutions really can not only have something aesthetically beautiful, which is it changes our understanding of our world and our place in nature, but it also can provide unexpected practical benefits. I can't imagine a more powerful or more beautiful statement on the importance of both curiosity-driven research and our evolutionary connection to the rest of life on our planet than that. Thank you very much.